Okay, so we're going to do genetic principles in blood banking. So genetic. Your blood group systems are groups of, what are they on red blood cell membrane?
What?
What is it that's on the red blood cell membrane?
Antigens.
Yep, antigens on red blood cell membrane. They share related serologic properties. So like everything in RH, in the RH group, shares serologic properties, and they also share genetic patterns of inheritance, and same for ABO. And then what's the biggest thing you think of first when talking about genetics?
What you get from your parents, your DNA.
Yeah, your DNA. So whenever someone says genetics, that's usually the first thing that that comes to mind. And so your DNA is like a blueprint for who you are. Now, your DNA is where the genetic material is found. Where the genetic material is. Now, I want to insert a little picture. So it goes macroscopically to microscopically. It goes from a cell to the nucleus of the cell to chromosomes, so pairs of chromosomes, to DNA, and then each DNA strand goes down to a gene, which goes down to proteins. They're made of proteins. So it goes from the cell to the nucleus. And then now we're going to go even deeper into the chromosome. Chromosomes. That's not what I wanted. There we go. Chromosome. There are 23 pairs. And how do you get your pairs of chromosomes?
I don't remember.
You get 23 from mom and 23 from dad.
Oh, right.
So that's where you get your pairs from. So 23 pairs from mom and 23 pairs from dad, and leaves for 46 total. And they're diploid, meaning they come in sets of 2. So there's two together.
What does diploid mean?
Diploid means that that there's 2 together. So like if you look here, there's 2 chromosomes together all the time. That's what it's talking about.
Okay.
And then what else do we have? We have genes. Our genes are further down the line. So they're even more condensed than the chromosomes, which are even more condensed than, I'm sorry, your DNA is part of them. I should switch this one. Let me fix this. You already know the nucleus and stuff, so I'll start with chromosomes. And so again, they're diploid. Sets of 2. And then they are, you've got 23 pairs. 23 pairs. So it goes from your chromosomes then to your DNA. Which is your blueprint. And then from DNA, it goes down one further to your genes. And what are your genes?
What you're inherited from your mom and dad?
Exactly. Yep, basic units of inheritance on a chromosome. And now the location are going to be, I'll put gene location so we know we're still talking about gene locations. Okay, so then you've got 2 choices for gene location. One is going to be cis, C-I-S, and those are alleles on same chromosome. Because how many do you have? How many chromosomes?
23.
23?
Yep, that's how many pairs. But how many does one pair have?
2?
2. So cis is alleles on the same chromosome, so not one on each, but 2 on the same one. And then your other one is going to be trans. And now your trans is going to be alleles on opposite chromosome. Which makes sense, because you know that trans, means to go across, right?
Mm-hmm.
Okay, now what's going to be our, let's see, next one is going to be alleles. Am I confusing y'all with the way that the bullet points are?
A little.
It's okay? Or you're under, each highlighted one is like pretty much starting a new bullet point.
Okay.
But I want to show like the breakdown. Does that make sense?
Oh, okay. I get it now.
So I wish I could. So like chromosomes is its own with the two little black dots, and then DNA is its own.
What I would do is an arrow.
Huh?
Wouldn't an arrow be more efficient?
An arrow would be better
I would think.
than
Like going from the chromosome to the genes to the alleles.
uh-huh. Well, I have it up here in the picture.
Yeah, that'll work.
Okay.
That's fine.
So then alleles are alternative forms of a gene. And they are found at each locus. And now the best way I can explain alleles is that they are like flavors of genes.
What is a locus?
We'll get there.
Okay. Oh, I guess. I do that for so long. It hurts.
Oh, I guess I can put it here. And then so your locus or loci are is like the site. And I'll talk about it a little deeper here in a minute. So the best I could find to explain the difference between alleles and like your loci is going to be your locus is the site at like which the where it would be on the chromosome, and the allele is your different flavors from that. So your locus would be like your favorite Tootin' Totem, or your favorite gas station where you go and get your drink every day, or your favorite coffee shop where you go get your coffee every morning. And then your alleles will be the alternative forms of drinks that you can get at that location. Does that make sense? So the locus is always the same on every different person. The site to where that gene is held is going to be the same, whether it's you or me or someone else or someone in another country. However, we all have different alleles at that location or site. And then multiple alleles at a single locus are called polymorphic. So your polymorphic is multiple alleles.
What did you say before that? Your polymorphic or?
Before polymorphic? Before polymorphic, I said that your alleles are like your flavors of genes. So if you went to
No, you were describing what polymorphic multi-allies are.
oh, they are multiple alleles at a single locus. So multiple alleles at a single site.
And what does polymorphic mean? I mean, just many?
Many alleles at a single locus. Okay, so let's say this is one of your chromosomes.
So that's a chromosome you drawed?
Yes, it's supposed to be anyway, but we know how my drawings go.
No, that's fine. Just making sure I got it down right in my
Yeah.
head. I'm just making sure I got it right in my head.
Yep, you got it. Oh, yeah, it comes out the best. Okay, so let's say this is a locus or site. So that's going to be don't worry, I'll fix it. No.
All
Okay.
right, I'm just no, you're fine. You're fine. You're fine. It's not that bad. It's not school. It's just tutoring. She thought she was interrupting something bad.
I know my dad came in earlier too. I don't know if y'all saw.
It's like it's okay.
Okay. So can you see the little triangle and the little diamond here?
Yeah.
If I made it bigger, I could have showed more. So the locus or loci is a site, and that's going to be same across every person. And then the ABO antigen or the ABO allele would be like whichever one is on that site. So let's say you were A, you might have a triangle, or if you were B, you might have a diamond. Does that make sense?
Yep.
You just have whichever one you receive genetically. So there's more variation for alleles. And like I said, your loci or site would be like favorite coffee shop. And then your ABO or allele would be your daily drink, or I guess, yeah, your drink of choice. Does that make sense?
Yeah.
Okay, and then next we have, I'm gonna go over that. There we go. Then we have a haplotype. Now, what's a haplotype?
Is it that same thing that when it needs like some certain thing in order for it to activate?
Nope,
It's
it's
like
a
a
little
key?
different. That was a, I know what you're talking about. It's a,
Captain or something like that.
oh, I know what you're talking about. I just can't think of it right now. It's a carrier in
The
immunology.
little haptin.
Haptens. Yes, haptens.
Yeah.
It's different than haptens.
Okay.
So your haplotype is a set of linked genes, and then your linked or linkage is when two genes close to each other are inherited together. Two genes close to each other are inherited together. Y'all following so far?
All right, so I get the two genes that are close to each other,
Uh-huh.
but are inherited together. I don't get the concept together.
So sometimes if there's a haplotype or a set of linked genes, this isn't like an exact example, but like, let's say if you have, if brown hair and brown eyes are a haplotype together, or like blonde hair and blue eyes, if they're a haplotype together, then if someone has brown hair, they're going to have brown eyes, or if someone has blonde hair, they're going to have blue eyes. So when they're linked together, that just means if you get one, you're going to get both.
So a person can't have brown hair and blue eyes?
Well, a person can, but not if they're not if it's a haplotype.
Oh, okay.
I just used that as an example. Let me see if I can find a real example. Can you all see this? The internet?
Uh-uh.
Okay, just a minute. So an example would be like your RH. So let me be RH. So your DC and E. All so if you so they're all going to be together. Now not all genes are haplotypes. It's just when they're linked. So it's inherited as a group from one parent. In blood banking, haplotype is usually used to refer to closely linked alleles inherited from one parent for a particular blood group. For example, in the RH blood group, the alleles for D and big C, big E, little e expression were previously thought to be passed from one parent to a child as a unit. So for RH, a child receives one RH haplotype from his mother and one from his father, and then the combination of alleles determines the RH antigens that will be expressed on his red blood cells. So instead of getting just one, I don't know, it's hard. I mean, they're just together. They're always together.
So if you have a haplotype, you're going to have a big C and a little c, or a big E and a little e. You're not going to have a C and a little e, is what it's saying.
Well, you could, but you're going to get a haplotype from mom, and you're going to get a haplotype from dad, and it squishes them together. But they're instead of getting like instead of getting like C from mom and D from dad and E from mom, you would get DCE from mom and DCE from dad, and then it mixes your together. So you know how you get let me see. Did that one make sense? That instead of getting like, if you instead of getting D from dad and C from dad and then getting your E from your mom, you get a DCE from dad and you get a DCE from mom, and then as a genotype, and then you phenotype whichever ones are dominant.
Okay, so you're going to get the same thing from mom and dad. It's just when you go from there, it could change and pair up a little different, is what I'm
Yeah.
trying to. Does it make any? I don't know if I'm making any sense.
Okay, we'll get through it and then see if at the end it doesn't get a little clearer. Okay, so now we're going to get into phenotype. So what is the phenotype?
All right, give me a minute. I know this. I don't know. I don't remember. did, but I don't remember. Where did I put it? You're fine.
Y'all remember what your phenotype is?
Mm-hmm.
What is it?
Physical attributes or what you see.
Yep. So what you see. What you see. So it's going to be your physical expression of traits. It's determined by hemagglutination of red blood cell antigens. What is that?
Determined by hemagglutination, RH, antigen, physical expression of traits. Could be that.
Determined by hemagglutination of red blood cell antigens using anti-sera. So what is that hemagglutination of red blood cell antigens using anti-sera? Does that sound like anything you know about?
By hemagglutination. Is that what we were doing in the lab that one time?
Yeah, so what do you do in the lab?
Determining what RH they have, I guess, and other things? Antigens and antibodies?
Which one determine antigens?
Let's see, one that. Oh, wait a minute. Using anti-sera. Oh.
That's going to be your forward typing. So determined by hemagglutination of red blood cell antigens using anti-sera is just a fancy way of saying you're forward typing. Because that hemagglutination is what all your blood bank tests are based on. Not all of them, most of them. Is what most of your blood bank tests are based on. So determined by hemagglutination of red blood cell antigens using anti-sera is just a fancy way of saying forward typing.
Okay.
So it's produced by your genotype, and we'll talk about that in a minute. But you have a genotype, and through that is how you get your phenotype. And then there's a phenotype calculation. And what that does is enables you to find a unit of red blood cells with certain antigen characteristics. So enables finding unit of red blood cells with certain antigen characteristics. And so when it's talking about certain antigen characteristics, are they going to want them to have an antigen or not want them to have an antigen?
Say that again.
When they're talking about certain these antigen characteristics, what do they mean? What kind of characteristics are they talking about? It's going to be antigen negative because you don't want them to have the antigen, right? That's what blood bank's all about. Not predisposing someone to make an antibody. There we go. So you're going to want these to be antigen negative. Does that make sense why?
No.
So if a patient has antibodies, then you're going to want your donor unit to not have any of those antigens. So when you do a phenotype calculation, you're just trying to see an idea of how many units you're going to have to test to find that, the amount of samples you need to for the patient that are antigen negative. Because what happens if a patient has antibodies and you give them donor blood who have antigens of the same kind of their as they have antibodies too?
They're gonna cross each other out, aren't they? They're gonna attack each other.
Yeah, they'll get a transfusion reaction. So the whole point of your phenotype calculation, let's say you need to go and get, we'll start off with one. You need 1 unit of blood for someone with a certain amount of antibodies already in their system. And so when you go get this, you need to know whether you need to grab 3 different choices of a certain blood type or 20 choices of a certain blood type. So that way you can sit down and test them all in one sitting. You don't test one, go get another one, test it, go get another one. So this calculation just kind of tells you, okay, I need to grab 5 different bags. I need to grab 10 different bags. I might need to grab 30 different bags and then come sit back down.
Okay.
Does that make sense?
Yes.
Okay, so how you do it is going to be, I'm going to show you on the like the class PowerPoint here in a minute, but I'll write out the calculation first. So it's going to be the decimal negative. Well, I'll put change negative percent to decimal. And then next, once you have your decimal, it will be your decimal negative. And then that one's going to be multiplied times your decimal negative of your next antibody. So I'll put that in here. Decimal negative of first antibody. Decimal negative of second antibody. And it'll be times the decimal negative of third antibody, and on until you, let's see, until you have all the antibodies that that patient has.
Wait a minute.
Okay, now let me show y'all on a PowerPoint what I'm talking about. Do y'all have this down so far?
Yeah.
Okay, so your phenotype calculation, this is a patient with multiple antibodies. So for this instance, they have anti-big C, anti-big E, and anti-big S, or S, and they need blood. So this is their known. So this is their positives, and then this is their negatives, negatives, and then this is their convert the decimal. So this isn't the decimal of their positives, it's the decimal of their negatives. So the percent negative, you change that to a decimal. So how do you change it? How do you change from a percent to a decimal?
Just move it back 2 places.
Exactly. And then what if they tell you she has big E positive 30%, but they don't give you the negative?
Then you would know it'd be 70. It'd be the difference out of 100.
Exactly. Okay, so then you've got your converted decimal. Now you take all of these. So, So in this case, there's just 3 of them. So you take the 3 of them and you multiply them times each other.
Okay.
And then, so that one's going to be 0.3, 0.7 times 0.45. And in this instant, it comes out to 0.0945, which they rounded to 0.1. So that's, what's 0.10 in a percentage?
10%.
10%. And then, so about 10% of blood will be negative for all of their antibodies.
Okay.
Antigens for their antibodies. Now, about 10% of the population will be negative for all 3 antigens. About 1 in 10 units will be compatible. So that 10% is the same as 1 in 10.
Okay. Give me a second. I'm going to write this down a little bit.
Okay, and I'm going to put a couple notes in here too. Okay, so once you continue until all patients' antibodies are accounted for. Together. %. So after all the antibody negatives in decimals are multiplied together, you change it back to a %. And that is going to be the % that they are negative for all the antigens. percent negative. Okay, so then after all the antibody negatives and decimals are multiplied together, you change it back to a percent, and that is the percent negative for all antigens that a patient has antibodies directed towards. And then you take that number, so and then you multiply. And then you multiply it. You multiply it by 100. Let me see. Let me show you, like, sorry, I don't know the best way to say it. So what I'm trying to get at is, like, to get that 1 in 10, 10 out of 100 can be reduced to 1 over 10. Or if it's 20, let's say your answer is 20%, then it would be 2 out of 10. If it's 23%, you would have, like, what, 2.3? You can move by 10.
So if it's 10%, you just take that 10% and times it by 10.
Yes.
That gives you 100.
Or no, I'm sorry, times 100. And then whatever. Let me find the actual, because I've never actually had to, like, explain the math for it. Because if it's 10%, it's 1 out of 10. If it's 20%, it's 2 out of 10. If it's 23, it's 2.3 out of 10. So I think you just divide it out.
So if it's 30, it would be 3 out of 10.
Yeah. Oh, there we go. Yeah, it's divided by 10. That's it. So it is the let me see if it was a decimal with it. Percent. I think it's divided by 10. So you have 10% negative. That means that 1 out of 10 patients And I believe we'll be compatible. So if you have 30%, 3 out of 10 patients will be compatible. 23%. How about that one for 23%?
So 35 would be 3.5 out of 10% are compatible.
What?
So 35 would be 3.5 out of 10 patients are compatible.
For 10%, how many out of 5?
No, 35%,
Oh, 35. Yeah,
3.5.
correct. And now that one was for multiple antibodies for 1 unit. Should have. Oh, they went down. That's why. So that one was in regards to multiple antibodies for 1 unit. So this is going to be multiple antibodies with multiple units. So the first step is going to be the same. You're going to get as far as you did on this one. And so we came out to so our answer was 10%.
Mm-hmm.
Now we need to take it further. So here they use a completely different one all together, but I want to do it for the one that we just did. So this is a patient with multiple antibodies and they need 2 units of blood. So what you do is you take your 10% and then you take 2 units needed and divide that by your 10%. But you have to do it as a decimal.
Which would be 5, wouldn't it?
You would take, just a minute, total. what did I put? That's the percent negative for all antigens. And then you want to convert it to a decimal. And then, so you take units of blood divided by your percent negative. And that is going to tell you how many antigen typing units you have to do or may be required to find the compatible units.
How many antigen what?
That's going to be how many units may be required to find your compatible units. So how many antigen typing you'll have to do to find the compatible units that you need. Approximate number of units. So it's going to be the units of blood divided by the percent negative will equal approximate number of antigen typing units you will have to complete to find the compatible units needed. So units of blood divided by the So do it for, let me share the other one again. So do it for this one. So if you have 0.10 or 10%, then what?
You divide that by 2?
Well, 2 divided by 0.10.
Wait a minute.
So your units will be on top. It's your units divided by your percent of negative.
20?
Yep. So you will have to use you will have to antigen type approximately 20 different donors to find your 2 compatible units.
What the heck? Let's see.
So 20 different donors to find your compatible.
Your 2 compatible units for this instant, yes. So what about if you this person needed 3 units of blood?
Give me a minute. 30?
30. So then you'd have to test approximate antigen type, approximately 30 units of blood to find the 3 compatible units that you need for the patient. So you don't really like need it like for patient health, but it helps to know approximately how many you're going to have to be checking to get there. So that way you can go grab, 3, or you can go grab, 10 and then at a time. Or I don't think anyone generally gets out like 20 or 30, but like there's a big difference in needing 5 out and needing 10 out, or needing 2 out and needing, 5 or 10 out. Does that make sense why this is
So,
done?
so what if they ask you a question like, how many units are you going to need for, or is that not
Like,
the,
I think just don't get the units confused. So about 1 in 10 units will be compatible. That is different than the 2 units needed to give to the patient. So each patient needs a certain amount of units given to them, and then but you're typing each unit.
okay.
And so your compatible units are different than the units you're given to the patient. So if they're like, how many units do you need to type to find a compatible match, it would be, your 22 or your 30, not 2. It'll tell you.
Okay.
It'll always tell you if the patient needs 1 or 2 or 3 units. But what you're usually trying to find is how many you have to generally type to find matches.
Okay.
Make sense?
Yes.
So which one goes on top when you have multiple antibodies and multiple units? When you divide? Yeah, your units of blood needed. So that's how many units are being given to the patient.
Right.
And I said how many will be given to the patient. They don't always like give them all. But for the sake of space on the paper, could be given. I guess I'll put it could. And then the percent negative. Or I'm sorry, it's going to be as a decimal though. Good?
Yes.
You too, Lily, good? Okay, and now before we get into like the genotype, I want to do a couple key terms. So the first one is homozygous. And so what this means is a person inherits identical alleles. And if you had a favorite coffee shop, would the alleles be the coffee shop or your drink?
The drink.
Yes. So these vary. So an example of these would be is group A, phenotype and genotypes is AA, or someone who is phenotypes is group B and genotypes is BB. And so what they do is they have a double dose of antigen. So your homozygous is a person that inherits identical, I wanted identical, not. Identical alleles. So they have a double dose of antigen. That means that there's antigen on these cells, and there's going to be antigen on these cells also. So it causes stronger reaction. And dosage is variation in antigen expression due to the number of alleles present. And so what that means is like, let me see, let me think about one that definitely shows dosage. I think your MNS are one. So your MNS antigens show dosage. Your RH shows dosage. KID, Duffy.
You said RH.
RH is one of them that shows dosage. So if you have big M, if you genotype is like big M, you're going to get a stronger reaction when you're doing your panels than you would if they were heterozygous. So like a big M and then a big N.
Say that very last thing you said.
That like on your panels is when this comes into play, and so it's going to be a stronger reaction if they're homozygous than if they were heterozygous. So let's say if it's heterozygous, it'll generally show like a 2+ or 3+. But if it's homozygous, it'll show like a 3+ or 4+ reaction. Ready, keep going. Let me put in a picture. And of course, I have an example. So here, they're talking about M and N, part of the MNS blood group right now. So which one would this be, heterozygous or homozygous? This line 1.
Oh.
I don't know.
Is it homo?
Why?
The 3+ antibody reaction?
Nope, you're not looking at that. That just tells you what your lab results are.
Well, let's see.
I'm talking about right here, right now. So
So wouldn't that first one be hetero and the second one be homo?
exactly. So this one has a positive and a negative, and then this one's got a positive and a positive. So this one's homozygous with 2 positives, and this one's heterozygous with a positive and a negative. So what that tells you is that's for M and that's for N. So if this N is negative, that means that that person has big M, big M. So it means they're phenotyping with the M, but that shows that negative N shows that they have they inherited 2 big M's, or 2 M's, which makes a stronger reaction. So that's a 3+ reaction compared to being, I'm sorry, I switched them for y'all, because normally if you're talking about a different one, that would be the case. But in the case of M and N, since they're separate antigens, if you have M but not the N, then that means you're homozygous for M. And then if you have an M and an N, you're heterozygous. I'm sorry, for this one specifically, I got them switched.
Let's say I get that part, but writing it down, it's so for the homo it's M and little n, and for hetero, I mean for homo it would be M, capital M and capital N, and for hetero it would be capital M and little n.
Okay, so for this one specifically, it would be your homozygous would be this one, because it has just the M. So they don't have N, so they're big M genotype. And then to heterozygous. And for heterozygous, the person inherits different alleles. And so in that case, it would be like AO or BO or AB. And so in this case, it would be dose of antigen. And so in this instance, they would have MN genotype, yielding a 1+ reaction. And so up here, you'd have an MM genotype yielding a 3+ reaction.
So you're saying that, so the one on the bottom with the 2 pluses isn't homo, it's hetero.
Yes.
But those
No.
are identical.
I'll be back. I gotta do
Yeah, but
something.
I wasn't paying attention that these are M and N. So M and N are different antigens, but they're linked together. So if you don't have an N, it means you have 2 M's.
Which are the same. Okay.
Yes, which make it the same. Sorry, I wasn't paying attention
Right.
to that part when I explained it first.
Okay. So for it to be homozygous, one of them has to be negative and the other one has to be positive. To be heterozygous, they both have to be positive or negative.
No, it depends on the antigen. It doesn't 100% work like that. But you have to have two. What defines homozygous is a person inherits identical alleles. So in this case, they don't have an N. We happen to know that M and N go together. So if they don't have an N, that means they have 2 of the M's. And that makes it homo. But that's not always, the case. I wouldn't say it by one's positive and one's negative.
You would say it as if it's positive, then that means it has the M. If it's negative, it means it doesn't have the N antigen.
That is true. So if it's positive, it has the N antigen. If it's negative, it doesn't have the N antigen. But homozygous is identical, and heterozygous means they have different. So in the case of like your group A, if they're homozygous for group A, then you know that they genotype as AA. And if they're heterozygous for A, then you know that they're going to genotype as AO or AB, but AB wouldn't be group A, it would be group AB.
So let me ask you a question. What if one of those were both negative?
If they were both negative, I think you have to inherit some form of M or N.
Okay.
I think it's a require, like, I think it always happens that you have one of them at least positive.
Okay.
But like if N was negative on both of them, then they would both be type large M, large M. So they'd both be homozygous, yielding 3+ reactions. Or if your M's were negative, then they would both be homozygous for N, also yielding 3+ reactions.
So yeah, if it was opposite and it said negative for M but positive for on that top one,
Uh-huh.
if it said negative for M and positive for, negative for M and positive for N, it would be NN.
Mm-hmm. Exactly. And so what kind of reaction would you get?
Homo,
No, what kind of reaction? What
oh,
grade reaction? Stronger or not?
stronger.
Correct. Kind of makes sense.
Yeah.
Now these Mendelian principles, that's just saying that genes are inherited in pairs. Now, so where do you get the pair from?
Your parents?
Yep, so one from mom, one from dad. And now there's a couple other words that we need to know before getting into things. So there's autosomal. Let me put these in the middle. Y'all know what autosomal means?
The same?
Autosomal. Nope, it's actually in relation to where it's inherited, what gene it's on, or what location, kind of.
Inside the body, inside yourself, something like that.
So it's inherited on the autosome. And now what that autosome is, means it's not on the Y. So it is not on the X, because you know we get one X and one Y, or XX.
So it's not on the X?
Correct. Or I'm sorry, not on the X or Y chromosome. Okay, so like you've got two chromosomes. So you get one from your mom, and then you get either an X or a Y from your dad. Or I'm sorry, you get an X for your mom, and then you get either an X or a Y from your dad. So it is not on the X or Y from your dad.
The X.
It's not sex linked.
So neither?
Well, it's on the one that's not, that doesn't define your sex. So it's pretty much on your mom's. And then there's going to be X-linked. And your X-linked are going to be mutations in the X chromosome. I'll leave it like this one. And then our next term is dominant. The gene is expressed over another. So brown eyes is an example. And then there's codominant. What does that mean?
Equal.
Yes. So equal expression of two different alleles. What's an example of that you know of? What's an example of codominant? That's going to be your blood group antigens. So like if you have type AB, you're going to show both A and B. Some cells are going to have the A antigen on their surface and some are going to have the B antigen on their surface. And so what's recessive?
Too much.
Well, it does have something to do with much, with how much. It has to be inherited by both parents to be expressed. So your example for that is blue eyes.
So both parents have to have blue eyes in order for you to have it?
Correct.
My parents don't.
Oh, they can carry it. They don't, I'm sorry, they don't have to both have it. They both have to carry the gene, but they don't have to both express it. So the gene must be inherited by both parents, but they have two genes, and the brown eyes is dominant, so it's kind of like this AO or AB, I mean, sorry, AO or AA. If they have like, we'll say it's B for brown eyes. So if they have BB, then that means they have brown eyes and they can only pass down a brown-eyed gene. But if they have like the example would be like AO, so if they had like big B, little b, and they passed on the little b, and then your dad was big B, little b too, he could also, there's a 25% chance that the child will have the recessive traits. Sorry, I didn't think that through when you asked it. You must get the gene. They don't have to both display the gene.
Okay.
So example 1 is autosomal dominant. And in this case, a single gene induces a phenotype. Oh, let me go ahead and put in the genotype now. I think I can do genotype before I do these. So what is the genotype? Are you still there, Lily?
Yep, I'm still here.
Okay. So what is your genotype? Anyone?
Nick, make it.
Yes. So your example for this would be AA versus AO. Would be genotype. And group A would be your phenotype. And you can only determine the genotype through molecular techniques. Or family testing. I'm going to put Punnett square definition up here too. A Punnett square is used to predict probability of offspring's genotype. Used to predict probability of offspring's genotype. And so what it is every possible combination of maternal and paternal alleles of a particular gene. Okay, so here's a Punnett square. Well, these are multiple examples of Punnett squares. So you line up mom on one side, dad on the other. Doesn't matter which way you do it. So if mom has the genotype of AB, what is she going to phenotype as?
Maybe.
Yep. And if let's say dad has type OO, what is he gonna phenotype as?
Oh.
Phenotype.
Oh.
O. Yep, just group O. So how about Michelle? Give me this one down here. Tell me the genotype and phenotype.
All right. So the genotype would be AO for the mother and AO for the dad.
Yep.
And phenotype would be oh god. I don't know.
So go back down here. So example, AA or AO would be the genotype, and group A would be the phenotype of AA or AO. So what is the phenotype? What shows? What will that person show in lab tests?
So the mom would show AO and the dad would show AO.
Nope, that's their genotype. What is their phenotype? Because here it says down here that you cannot, the only way you can determine the genotype is by molecular techniques or family testing. So you can't tell the genotype by forward and reverse typing, but you can tell the phenotype.
Would it
So
be A?
yeah, group A. Exactly. So the phenotype would be A, but the genotype is AO.
So the phenotype is A, but the genotype would be AO.
Yes.
Okay.
Technically, the phenotype is group A, because that's what it shows.
Okay, yeah.
And then the genotype would be your AO. Lily, how about you do this one?
That one in the corner. Okay.
The middle left one.
Okay. Let's see. The genotype would be AO and OO,
Yes,
and the phenotype would be A and O.
correct. So now that's your Punnett square. I moved it up here with the like terms, some of the key terms. And then now I'm going to go back down to the genotype again.
Can you go back up to that Punnett square real quick? I didn't. Okay. All right. Thank you.
Okay. Uh-huh. Okay, so now if something is autosomal dominant, so I took it's either autosomal or X-linked or sex-linked, and then it's either dominant, codominant, or recessive. So this one's autosomal dominant. So what that means is a single gene induces a phenotype. Single gene induces phenotype. So what does that mean?
Yeah.
Anyone have a guess?
I'm not sure.
Okay, so autosomal dominant means one single gene induces the phenotype. So if we're talking about AA or AO, so the A would be dominant because if you have it even with an O, then it will still induce a phenotype. So the person, what they look like, what they group as, will still be group A, even though they have an A and an O. So your A is dominant and your O is recessive. And then, so you need at least one affected parent. So one parent has to be affected if you get a dominant gene. Children have a 50% chance of being affected. And then males and females are equally affected. And you've got autosomal recessive. So what's recessive?
They have to be inherited by both parents.
Not quite. That's that's going to be your Mendelian principles and codominantly. Specifically recessive.
Up there where we put the definition for recessive, you put genes must be inherited by both.
Yes. But it doesn't have to do with like getting one from mom and getting one from dad. You have to get both, the same one from mom and dad. So it's not just about getting one from mom and one from dad. It's getting the same one from mom and the same one from dad. So 2 genes are needed to induce a phenotype. It's located on the autosome. So that's your non-sex linked gene. And let me see if I can't find a better way to explain the sex-linked versus non-sex-linked. Yeah, autosome is just any chromosome that's not a sex chromosome. So you get, okay, so out of your 23 pairs, 22 of those pairs are autosomes, and then one of them is your sex-linked chromosomes. Let me go up here. Autosome. So individual has So an individual has 22 pairs of autosomes out of your 23 pairs total. And then your extra chromosome is going to be your X-linked or sex-linked chromosome. So you'll get an X from mom, and then dad will either give an X or a Y on that last chromosome. Does that help?
Yeah.
Okay. So your autosomal recessive, you need 2 genes to induce the phenotype. They're located on autosomes. Now, carriers. Remember we talked about carriers in our hemophilias mostly? What that's talking about is the dominant gene masks the recessive. So it means that, like, up here, this person is a carrier of type O blood, but the type A is dominant, and so they phenotype as type A, but they are a carrier of O. So if you come up here and you've got 2 parents that are AO, your offspring have a chance of either being AA, AO, AO, or OO. So there are, it's a 25% chance that they are homozygous for A, 25% chance that they are homozygous and for the recessive trait of O, and so they will be group O. And then there's a 50% chance that they are group A and carriers of O. Make sense?
A little.
Okay, I'll keep going, and then afterward, if there's any questions, you can ask away. Okay, so you can pass a recessive trait to a child. Carriers can pass recessive trait to child. There's 25% chance that the child is affected. And an individual may have unaffected parents. So what this is getting at is that, let me crop out the one I'm referring to here. Well, I'll show you them all first, and then I'll crop it. Okay, so for autosomal recessive, you need both genes to induce a phenotype. So this group O right here is recessive. So you need both genes to be O to phenotype as type O. So out of all these squares, the only way to get a group O child is to have a either homozygous parent for group O or a heterozygous parent for another blood group, and a homozygous for, or two heterozygous. You cannot have either parent be homozygous for A or B and have a group O child. So when you have a carrier, that means like here, where you have group A phenotyping, but they are actually have a genotype of AO. So if you look down here, this 25% chance is talking about this for recessive. And then the other example would be of autosomal dominant, would be like this one here, where you have one parent that has the dominant gene, so at least one affected parent. So this one's recessive, and this one is an affected parent, and so they have a 50% chance of typing as group A. See? And then these are going to be both group O.
So what if both parent is OO?
Then your only choices are going to be OO babies. Every child they will ever have will be the recessive OO genotype and group O phenotype.
Okay.
And then we talked about now our autosomals, and now we're going to do our X-linked or sex-linked. And I started there with dominant, so I'll start here with dominant. And what that means is all female offspring of affected males will be affected. So all female offspring of affected males will be And so all female offspring of affected males will be affected. No male offspring of affected males will be affected.
Uh-uh.
An affected female has a 50% chance of having an affected child. And then all affected individuals have one affected parent. So whenever you're talking about dominant, you know that you have one parent that is visibly showing the trait. And then we have X-linked recessive. Primarily males are affected. You have obligate female carriers. So what does that mean? That means that all female offspring of affected males will be a carrier. Male offspring of affected males are not affected. And that is because they don't have a Y. Or I mean, they get a Y, so they don't have it. If it's X-linked, it's on the X, and so if they have a male, they get the Y instead of the X, so they're not going to get it. Does that make sense?
Mm-hmm.
And then males only need one copy to show their phenotype. And females need 2 copies to show her phenotype. And then once y'all catch up, I've got an example.
Okay.
So most blood group system genes are on autosomes, except those of the XG system. Now, those genes are found on the X chromosome. So, if the father carries the XG allele, he will pass it to all of his daughters, but not to any of his sons. Why is that? If the father carries it on the X chromosome, he will pass it to all of his daughters, but not any of his sons.
Because he doesn't have the Y chromosome?
Yes. So all of his daughters will have the X chromosome where it's located, but none of his sons will have that X chromosome because he gives them the Y. And so it's not on that chromosome, so they're not going to have it. Now, if the mother carries the allele, not the father, all of their children will express it. Why? What gene does mom give?
X.
X. So all of her children will express it.
Cool.
Questions? Okay, that's all I got for this one. And I'm going to make a short video on the, what is it going to be? On that Hardy-Weinberg formula. So I'll do one on the Hardy-Weinberg formula and make it separate.